Welcome to Biochemistry of Metabolic Processes. Today, we're going to explore one of the most powerful energy-generating pathways in the human body, the beta-oxidation of fatty acids. We will discover how your cells chop down massive fat molecules to keep you alive and energized, especially when you are fasting or exercising. To understand why this process is necessary, imagine you are trying to keep a campfire burning through a long, cold night. You have a massive tree trunk, but you cannot just throw the entire trunk into the fire because it is too big to burn efficiently. Instead, you take an axe and chop the trunk into smaller, evenly sized logs. In your body, a fatty acid is exactly like that massive tree trunk, storing a huge amount of potential energy. Beta-oxidation is the biological axe that systematically chops this long fatty acid chain into smaller, manageable pieces that your cells can actually use. Now that we have our analogy, let us look at the biological reality. Beta-oxidation is a fundamental catabolic pathway that takes place inside the mitochondria of eukaryotic cells. The process gets its name because the chemical cleavage occurs at the beta carbon, which is the second carbon atom away from the functional carboxylic acid group. During each cycle of this pathway, a 2-carbon fragment is removed from the fatty acid chain and released as a molecule called acetyl-CoA. Alongside this physical chopping, the process also extracts high-energy electrons, which are captured by the carrier molecules NADH and FADH2 to drive massive ATP production. Understanding this pathway is absolutely critical because it explains how our bodies survive when carbohydrates are not available. During periods of fasting, intense exercise, or starvation, your body turns to its fat reserves. It breaks them down through this exact pathway to keep your vital organs functioning. For instance, your heart muscle relies almost exclusively on the continuous beta-oxidation of fatty acids to maintain its rhythmic beating day and night. If this metabolic pathway were defective or missing, a person would suffer from severe energy crashes, muscle weakness, and potentially fatal heart complications during even brief periods without food. Since we know how important this process is, we need to calculate exactly how much energy we get from a single fatty acid chain. This brings us to our first formula, which determines how many cycles of beta-oxidation are required to completely break down a fatty acid. The formula is C equals N divided by 2 minus 1. Here, C represents the total number of beta-oxidation cycles, and N represents the total number of carbon atoms in the even-chain saturated fatty acid. If the value of N increases, meaning the fatty acid chain is longer, the number of required cycles will naturally increase as well. Before we move on, let us see this formula in action with a concrete example. We will calculate the breakdown for palmitic acid, a very common saturated fatty acid in the human body that contains exactly 16 carbon atoms. We're given N equals 16, and we need to find the number of cycles. We substitute 16 into our formula, dividing it by 2 to get 8. Then we subtract 1. This gives us a final answer of 7 complete cycles. Physically, this means the biological axe must swing exactly 7 times to completely chop up the 16-carbon chain. Now that we have used this formula, let us understand where it comes from conceptually. Why do we divide by 2, and why do we subtract 1? We divide N by 2 because each cycle of beta-oxidation chops off exactly 2 carbons at a time. However, As we chop down the chain, we eventually reach a final piece that is exactly 4 carbons long. When the biological axe swings that very last time, it splits the 4-carbon piece into 2 separate 2-carbon pieces simultaneously. Because we get 2 pieces from one single chop, we do not need an extra cycle for that final piece. That is why we subtract 1. So, we have proven how to find the number of cycles. This brings us to calculating the actual products generated. Our next formula determines the total number of acetyl-CoA molecules produced at the end of the entire breakdown process. The formula is A equals N divided by 2. Here, the variable A stands for the total number of acetyl-CoA molecules, and N is again the total number of carbon atoms in the original fatty acid chain. Because every single acetyl-CoA molecule contains exactly 2 carbons, we simply divide the total carbon count by 2. Notice that the number of acetyl-CoA molecules is always exactly 1 more than the number of cycles. Let us apply this new formula to our previous example of palmitic acid. We are again given a total carbon count of 16, and we are asked to find the total number of acetyl-CoA molecules produced. We chose this formula because it directly relates the starting carbon chain length to the final 2 carbon products. By plugging 16 into our formula for N, we simply divide 16 by 2. This gives us a final answer of 8 molecules of acetyl-CoA. Physically, this means our 16-carbon tree trunk has been successfully chopped into 8 perfectly sized 2-carbon logs, ready for the cellular fire. Now that we know how many logs we have, we must account for the sparks generated during the chopping process. Alongside acetyl-CoA, beta-oxidation extracts high-energy electrons. The biological rule is that each individual cycle produces exactly 1 NADH molecule and exactly 1 FADH2 molecule. Therefore, if a fatty acid requires C cycles to break down, it will generate exactly C molecules of NADH and C molecules of FADH2. Returning to our palmitic acid example, since it went through 7 cycles, it generates exactly 7 NADH and 7 FADH2 molecules in total. This massive yield of products explains why fats are such an incredibly dense and efficient source of stored energy. This brings us to the ultimate goal of the pathway, calculating the total cellular energy produced. We can express the total ATP yield as a comprehensive formula. Total ATP equals the number of acetyl-CoA molecules times 10, plus the number of cycles times 2.5 for NADH, plus the number of cycles times 1.5 for FADH2, minus 2. Each acetyl-CoA gives 10 ATP, each NADH gives 2.5 ATP, and each FADH2 gives 1.5 ATP. Finally, we must subtract 2 ATP equivalents at the end. This subtraction represents the energy consumed up front to activate the fatty acid before it can even enter the mitochondria. Let us put everything together to find the total ATP yield of stearic acid, an 18-carbon fatty acid. We are given N equals 18. First, we calculate the cycles, which is 18 divided by 2 minus 1, giving 8 cycles. Next, acetyl-CoA is 18 divided by 2, giving 9 molecules. We plug these into our ATP formula. 9 acetyl-CoA times 10 is 90 ATP. 8 NADH times 2.5 is 20 ATP. 8 FADH2 times 1.5 is 12 ATP. Adding these gives a gross total of 122 ATP. Finally, we subtract the 2 ATP used for activation. Our final answer is a massive net yield of 120 ATP molecules, providing enough energy for prolonged physical activity. Now that we have calculated the energy yield, let us visualize the actual chemical machinery. What you are seeing here is the elegant, repetitive four-step cycle of beta-oxidation. Notice the long-chain fatty acyl-CoA entering at the top. The cycle consists of four distinct reactions. First, an oxidation step that generates FADH2. Second, a hydration step where water enters. Third, another oxidation step that generates NADH. And finally, the fourth step, cleavage or thiolysis, where a molecule of coenzyme A enters and our 2-carbon acetyl-CoA is released. The remaining shortened fatty acid chain then loops back to the top to undergo the cycle all over again until the entire chain is consumed. We have reached the end of our journey through beta-oxidation. Let us reinforce the most important ideas. First, beta-oxidation is the essential metabolic pathway that systematically chops long fatty acid chains into 2-carbon acetyl-CoA units. Second, we can mathematically predict the breakdown using the formula C equals N divided by 2 minus 1. Third, alongside acetyl-CoA, every single cycle extracts high-energy electrons captured by NADH and FADH2. Finally, this process generates a massive amount of ATP, which is exactly how our bodies, and especially our heart muscle, survive between meals and during intense exercise. You now fully understand the incredible efficiency of human fat metabolism.